So we've got three speakers in this session, and the first one I'd like to introduce is Nadav Elad, who did his PhD with Helen Sable here in the UK in Birkbeck, and then returned to Israel to work with Ohad Medalia before Ohad moved to Germany. And then after that went on to become a staff scientist at the Weizmann Institute. So I'd say Helen and Ohad both obviously have done a lot of work in tomography, although Nadav highlights to me that Helen wasn't working on tomography when he was working with her. And then he went on a sabbatical to EBIC a couple of years ago to really learn the ropes of tomography. And he's going to be giving a talk now about his work there on rubisco from the pyrenoid in chloroplasts. So over to you, Nadav.
Thank you, Morgan. Is that working now? Yeah, you can hear me. Yeah, that's all true. So, so I'm a staff scientist at the EM unit at the Weizmann, and I had that privilege 2 years ago already, at 2023, in the spring of 2023, to visit EBIC for 3 months. That was a very nice experience. Pejun hosted me along with all the nice and wonderful EBIC staff. And this is the work that came out of it. By the way, this is the Harwell countryside. If you look outside of the ring, you know, then yeah, it's very beautiful in the spring. Yeah. So rubisco, as you may have heard, is the most important protein on Earth. So it's important because it catalyzes the fixation of inorganic carbon, and the vast majority of CO2 is indeed fixed by rubisco. It's a major player in what's called the Kelvin cycle, and the energy comes from the light harvesting complex from the sun mainly. So this is all done in chloroplasts, but also outside of chloroplasts sometimes. Rubisco has this carboxylation reaction, which is favored, and this fixes the CO2 and assimilates the carbon into the central metabolism. So you get the initial precursor for sugars and almost all the biomass. However, it also has a competing reaction, a competing catalytic reaction, which is called oxygenation. And this is where this ribulose biphosphate interacts with O2, with oxygen, and then you have actually energy loss and loss of mass. So cells would like to avoid this, obviously. It's also, rubisco is very slow in its catalytic rate, and therefore you need masses and masses of it in order for cells to grow. And in order to do that, to accomplish this, to have rubisco mainly doing the carboxylation reaction rather than the oxygenation reaction, different organisms developed what's called CCMs, or CO2 concentrating mechanisms, where the CO2 is pumped into an organelle, and the surrounding of rubisco is enriched with CO2. Such an organelle occurs in Chlamydomonas. It's called the pyrenoid. It's not a membrane bound. It's just a condensate or phase separated organelle packed with rubisco, and CO2 is pumped in order to keep it running at full velocity. So pyrenoid is a really important organelle, as 30% of global carbon fixation is mediated by pyrenoid. So it's really worth investigating. So yeah, I arrived in EBIC to learn the cryo-FIB and cryo-ET, subtomogram averaging, and so on. Yeah, initially they put me on the SIOS doing manual milling from morning till night, and then we could get some time on the very busy Aquilos I'm not complaining or anything. That's it was a good experience. Yeah. And then could get some from Pejun's lab, I should say, could get some time on the Aquilos for more automated milling, and also Mod helped us with milling on the Arctic. So we got enough lamella, and we did and then we did the cryo-ET. Here you can see the pyrenoid, right, with the rubisco inside, starch granules that surround the pyrenoid, and some thylakoid membranes where the light harvesting complex sits. We'll concentrate here on the pyrenoid, and it's packed with rubisco. So this really looks like an ideal subject for cryo-ET, right? You can get as many particles as you want. However, if we looked at the literature, and it's on one hand, it's very easy to get rubisco, to purify it, and to get high resolution structures of it. It has been done for decades by crystallography, and also more recently with a single particle. And yeah, it's rock solid, so you can get it very nice structures of it. However, cryo-ET is looking at it in situ by cryo-ET appears to be much more challenging. So these structures are from the Ben Engel lab, and yeah, they highly, well, this one highly resembles the in vitro structure. However, still it's not high resolution, and the question is why. So why are we not getting high resolution? Where is the variability coming from? Is it very different, or what's going on there? So spoiler, I didn't get much better resolution than that, but we learned something on the way. So this is what I want to tell you. Okay, so the pipeline was pretty standard. Yeah, Crayola was quite instrumental in picking the particles. After intensive annotation, it could find the particles very reliably. And then we use Reliant 4 for most of the STA part. And indeed, what I found is that it's massively heterogeneous in the cell. There is also a limit in signal to noise that I'll talk about a bit later, but it's in first sight, it's massively heterogeneous, and I could only do the classification in C1, get meaningful classification in C1, and here you see the best, well, 20 classes, some results better than others, and I did 2 refinements, one in C1 and one in D4 of the same classes. Some result better than the others. The best resolution I could get is this class 12 that was resolved to 8.1 angstrom. And so with less than 10, better than 10 angstrom resolution, we see all the helices, all the beta sheets, the loops, and so on. So we could do some flexible fitting. This was done by Alireza Ramezani and Juan Parilla from Delaware University. And yeah, they did a really good job. A few words about the reaction cycle or the conformational changes in rubisco. So what you see here is a dimer. This is the functional dimer. So this is the two large subunits that are stacked once against the other. And in the interface here are 2 catalytic sites. This is the transition state analog from the crystal structure. And you have this mobile loops. The whole thing doesn't move. So the whole frame is super rigid. There are hardly domain movements there, according to the in vitro studies. And however, these mobile loops are very mobile, and they open and close during the catalytic cycle. So we overlaid the model that we got with the published crystal structure. It overlays very nicely. And what we can see that it overlays better with the closed conformation. The loops, the important loops overlay better with the closed conformation. And from that, we conclude that at least this class 12, the best resolved one is in a closed conformation. Then the rest of the classes were resolved to less than 10 angstrom resolution. So we couldn't do this modeling, but nevertheless, we could do this difference maps based on the thresholding, the false discovery threshold that Ariane was talking about. So this, for example, is a difference map between the C1 map of class 12 and the D4 refined map. So between the asymmetric and the symmetric one. And what you can see is here in green is the difference density. And what you can see that it nicely aligns to the interface and to the binding site of this EPY-C1. This is the second most abundant protein in the pyrenoid. It has this repeating binding motifs in between unstructured segments, and that what forms the condensate, the pyrenoid condensate, as can be seen illustrated here. Okay, so we see evidence for this, for the binding of this EPY-C1. However, it binds asymmetrically. So yeah, here, for example, we didn't find density. Okay, so and then we went on to characterize the best resolved classes. We couldn't characterize all classes, but at least the best resolved ones. These are class 7, 10, and 13. And these are their resolutions. If we just dock in the domains and do rigid body fitting, there is no movement as expected, as predicted. However, if we do this now, this trick of difference maps, what you can see here, so these are differences, for example, here between class 7 and class 12. This is the best resolved one. And class 7, 10, and 12, and 13 and 12, the blue density is the positive and the green is the negative, so the reverse subtraction. Okay, and what you can see is that they are substantially different from the class 12. So actually the best resolved class is the least representative, you could say. So the high resolution one is also not the least interesting one. And the main differences occur near the catalytic site. So this may be due to opening of this dynamics of the loops, but also maybe binding proteins that can bind there, and possibly neighboring rubisco molecules, complexes that touch each other. We also have some evidence for that. Another hotspot for differences is this interface between two functional dimers. So you have in a rubisco 4 functional dimers, right? And then the interface between them is, we see also major differences over there, specifically here, for example, here. And these, this is a bit speculative, but these correspond to binding proteins from bacterial carboxysomes that bind rubisco. Okay, so these are very different species, and in pyrenoid rubisco, there hasn't seen any binding over there at that interface. However, we do see evidence that there is something going on there, some proteins that bind there. Okay. And finally, Jens did some mapping back and to see the distribution of the different classes, and did some nearest neighbor analysis. And yeah, you can see that this is the main peak for the nearest neighbor. This was already published. However, he also saw some differences in the distances between classes. Okay, and also, yeah, and also here, so there is some deviation in the distances between neighboring according to the classes, neighbors according to the classes. He also saw that there is some difference in the distribution according to the concentration of CO2 correlated with the concentration. So next to the pyrenoid tubules, what's called, you can see them in the tomogram. There is a high CO2 concentration, which gradually reduces through the pyrenoid. And some classes, according to this, some classes tend to occupy this region more than others. Okay, so to conclude, we see variations in localized variation in pyrenoid rubisco, corresponding to local dynamics and maybe some binding proteins, but we don't see major domain movements. We resolved in a closed conformation active state, and we see evidence for differential distribution. And yeah, maybe the main conclusion here is that we need higher signal to noise in order to in these tomograms in order to see the finer details. This is a bit disappointing, but yeah, this is the way to go. Okay, so just I'll thank the people involved. The Rubisco project, of course, Pejun, Jen, Maud, Juan, and Alireza, and the wonderful and very generous EBIC staff that hosted me and shared all the knowledge, and I had a really good time there. And thank you for listening.
Thank you, Nadav. Any questions from the audience? Tom Burnley, are you here with your tablet?
Hi.
Hey.
Can you please tell us more about your difference map? Have you applied any weighting or scaling factors?
Yes, of course. So all the maps were filtered to the same resolution and then thresholded to this 1% false discovery density according to the paper from Carsten Sachse. And then these were compared, and the subtraction was done on this false discovery rate maps.
Just a reminder, if you're a remote attendee, feel free to post questions on Slack. Any other questions from the audience? Nadav, I was curious, it reminded me actually of Johanna's talk yesterday about finding patterns in the crevices in the ice. And I felt that I could see a matrix in your rubiscos. There was some sort of higher order structure. Am I just seeing things?
Yeah, there was a previous paper that shows that there is no such, so maybe very locally we could see some clustering with some pattern, but long ranges, it's a liquid-like arrangement. Yeah, and this was, yeah, this is quite evident.
Thank you. Anyone? Question? Wanda?
I was wondering, you mentioned photorespiration in the beginning. Do you think the arrangement and the organization of the pyrenoid is different under photorespiration conditions? Especially these pyrenoid tubules that bring in the CO2, does that change? And yeah, the rubisco arrangement and organization?
We haven't checked any other conditions, CO2 conditions from the literature. I don't remember if anyone has done this, looking at the morphology of the pyrenoid under O2. I mean, Yeah. No, so actually it condensates with high O2, with high oxygen. So it forms under high oxygen and disassembles under high CO2, right? So yeah, but we haven't checked the changes in morphology.
Any more questions?
That's a very nice talk. So you mentioned in the last, the only way forward is the increased signal to noise ratio. But I mean, given that so much variability, so how would you do that? Because adding more particles won't help.
Yeah, that's a good question. So the main noise is coming from the thickness of the sample, the ice and all the matrix. So you would need to thin. I guess that's the only option. However, when you thin down, you also get the damage from the gallium beam. So maybe writing it, or as Andreas, or something. Yeah, you need to do something more drastic in order to get the very thin layer to get to higher signal to noise. Or yeah.
Okay, in the interest of time, I think we better move on. So let's thank
Thank you very
Nadav once again, please.